Recently, one of our studies on a phase 2 single agent, single arm study evaluating ibrutinib in about 30 patients with previously untreated Waldenström Macroglobulinemia was published in the Journal of Clinical Oncology. This was a very interesting study. We felt that it was necessary to do this study because the approval by the FDA that was given in 2015 for ibrutinib in Waldenström's, it didn't really give any regard into if the patient needed to be treated or untreated to begin ibrutinib. Also, there was not really data on previously untreated patients. So we felt it was a scientific gap, and we designed a study to be able to identify these patients and treat them and see how they did. So we put about 30 patients on this study based on our previous experience that patients without NYD88 mutations did not benefit much from ibrutinib. We did not include any NYD88 negative patient on this study, so all our patients have NYD88 disease. What we saw in this study is that has been very beneficial to patients. We did not see any difference in terms of how well patients did compared to previous studies on patients who were previously treated. There were 2 previous studies in patients previously treated. One of them is from our group, the one that got actually ibrutinib approved by the FDA. And there was a multinational study as well in patients who were rituximab refractory. So those 2 studies were in previously treated patients. So the innovation in these studies, this was done in patients who were previously untreated. The reason we decided ibrutinib was an important molecule to test in Waldenström Macroglobulinemia is because of the finding of the NYD88 mutation. So what happens in a normal patient, a normal individual, is when the patient gets infected by a virus or bacteria, the NYD88 receptor gets activated, and then it couples with itself in the membrane of the cell, and it activates a series of reactions in the cell. One of them is the activation of BTK. And then it really occurred to us that a BTK inhibitor would actually be very important in patients with Waldenström's, because in patients with Waldenström's, the mutation in NYD88 allows NYD88 to activate itself, even in the absence of any type of trigger. So those cells are activated inherently all the time. And then that was the reason. So ibrutinib is a BTK inhibitor, and by blocking BTK, we were able to show, at least in the laboratory, that all the reactions downstream in terms of activation of survival and nuclear factor kappa B and other factors was clearly blunted by this inhibition. And so that gave us the idea that in the clinic this could potentially work.